Dear students, welcome to our program of biology for 3rd secondary. I hope you understand the lesson today, and I'm pleasure to introduce the biology syllabus for you. We are going to complete the last chapter of our curriculum today, inshallah. Let's go. We are still in Unit 2, Molecular Biology. But we have a new chapter today. The chapter is talking about nucleic acid and protein synthesis. Before explaining the lesson, I want to remember you what is the meaning of protein. Do you remember what are protein substances? From secondary 1, proteins are chemical substances in the living organisms' bodies. So the proteins exist inside our bodies. And they have another name. They are called biological macromolecules or polymers. Polymers mean that they are made up of many smaller molecules are called monomers. And in the case of protein, the monomer is amino acid. And we are going to know now what is the construction of protein, our structure of proteins. Number 1, amino acid is the building unit of protein that it consists of a carbon atom attached to the following. Number 1, a basic amino group, NH2. Number 2, an acidic carboxyl group, COOH. Number 3, a terminal group that is symbolized by R, which differs from an amino acid to a terminal group that is symbolized by R, which differs from an amino acid to another, because we have different types of amino acids. And the last component of amino acid is hydrogen atom. This is the chemical formula of amino acid. Consists of a carbon atom and is attached to amino group and carboxyl group and hydrogen atom and the terminal chain or the side chain that is differ from amino acid to another. We have different types of amino acids, about 20 types of amino acids that are called monomers. Do you remember? All 20 amino acids have the same basic structure, the chemical formula structure of amino acids, except for glycine, which has a second hydrogen atom joined to the carbon. And the other 19 amino acids have a fourth group, R, that is called also terminal group, which differs from one amino acid to the other. This is the formula structure of glycine. Carbon atom here is attached to 2 hydrogen atoms, not like the other amino acids that have a terminal group differ from amino acid to another. And this is the chemical formula for the 19 other amino acids. The repeated units of amino acids are linked together to form proteins, where when 2 amino acids combine together in a dehydration reaction. Do you know the meaning of the word dehydration reaction? Means removing of water molecule. A dipeptide compound is formed. This means when 2 amino acids bind together, form a dipeptide compound. For example, we have here 2 amino acids, and by elimination of water, a dipeptide will form. The combination of several amino acids together in dehydration reaction produces also a polypeptide chain. And this figure represents combination between several amino acids together and form polypeptide chain. We have 20 types of amino acids, and the difference between different proteins are due to the following factors. Number one, the differences in the numbers, kinds, and arrangement of amino acid in polymers. We said before, when two amino acids bind together, they form a dipeptide by elimination of water. So we have here two amino acids only, and the type of protein here is dipeptide. So when two amino acids combine together to form dipeptide chain. But here we have several types of amino acids, and they form polypeptide protein. So the number of amino acids is a factor that causes the difference in proteins. Polypeptide chain has more than two amino acids. So again, the number of amino acids is a factor that cause the difference in protein. Number two, this figure represents that when amino acids bind together, form a polypeptide, and the polypeptide form a protein, and the protein has 20 amino acids. The protein has 20 amino acids. We have different types of amino acids that we said before. The number and the kind of amino acids are factors that cause the difference in proteins. This amino acid is called glycine, and we have another type. We have alanine. It's another type of amino acid, and valine, it's another type of amino acid, and we have 20 types of amino acids. The factor number two is the weak hydrogen bond that may give the protein molecule its special shape. So we have two factors that cause the different types of protein. Number one, again, the number of amino acids, the kind of amino acids, and their arrangement. Number two, the weak hydrogen bond that may give the protein molecule its special shape. There are enormous variety of proteins in living systems and can be divided into two main groups. Protein is divided into two main groups. Number one, structural protein. These are proteins that constitute building materials in the living organisms, يعني help in the building of living organisms tissues. Number two, regulatory protein. These are proteins that regulate the numerous vital processes and activities for organisms. When you see the word vital processes, what the meaning of this? Like digestion, excretion, respiration. Protein regulate all vital processes inside our body and living organisms' bodies. And we are going to talk about structural protein. We have different examples of structural protein. We should to know first that the major types of fibrous proteins that act as structural elements. Do you know the meaning of fibrous protein? We are going to know. This figure represents the shape of fibrous proteins, and they are long filamentous protein molecules. Fibrous proteins are only found in animals. Examples of fibrous proteins: actin and myosin, used to construct the muscles and contractile system. Number two, This figure represents the muscular system that's inside our bodies, and it has actin and myosin protein. Number 2, collagens that form connective ligaments within the body. The ligament is the fibrous connective tissue that connects bones to other bones, and the ligaments between blood and lymph cells. This figure represents of collagen fibrous protein that exists between or in the ligaments between bones or another organs. Keratins that form protective covering such as skin, hair, hooves, horns, and feathers. So hair, skin, nails consist of keratin protein, and hooves, and feathers, and horns. Regulatory protein. These are the proteins that regulate the numerous processes and activities for the organisms. And we said processes like digestion, respiration, excretion, and any vital processes inside our body. Examples of regulatory proteins: enzymes. Enzymes are proteins, and this figure represents the shape of enzyme. They modulate or catalyze the chemical reactions inside the living organism body. Antibodies. Have you ever listened about the antibodies? They are the immune system inside our bodies that provide immunity against infection. What's the meaning of this? Antibodies defend the body against microbe, so protect the body from diseases, and antibodies are formed from proteins. Hormones and various hormones and other substances make each life from respond to the constantly changing internal and external environment. So proteins are very important substances that inside our body. Do you know what are food that have protein? Any milk products have protein, and all kinds of meat have proteins. So you should eat food that have protein. Proteins are called macromolecules or polymers, and the amino acid is the building unit of protein, and it's called monomer. And we have 20 types of amino acids, and we have 2 types of proteins. Do you remember what are they? Structural protein and regulatory protein. Structural protein that inside the living organism's tissues, but regulatory protein that help your body to perform all vital processes. Talk about ribonucleic acid or RNA. But before explaining the RNA, I'm going to remember you what the meaning of nucleic acid. Do you remember what is nucleic acid? They also called biological macromolecules or polymers. Polymers mean made up of many smaller molecules. They are called monomers. But in this case, the monomer is nucleotide. And we are going to know what is the molecular structure of nucleic acid. Nucleotide is the basic building unit of the nucleic acid. And the nucleotide consists of the following. Number 1, a pentose sugar molecule that is contain 5 carbon atoms. Number 2, a phosphate group is connected to the carbon atom number 5 of the sugar molecule. Number 3, a nitrogenous base that is connected to the carbon atom number 1 of the sugar molecule. And this figure represents the structure of nucleic acid. As you see, consists of pentose sugar that has 5 carbon atoms, 1, 2, 3, 4, 5, and a phosphate group that is connected to the carbon atom number 5, and a nitrogenous base that is connected to the carbon atom number 1. We have 5 types of nitrogenous bases, which are adenine and is symbolized by A, guanine that is symbolized by G, uracil that is symbolized by U, cytosine that is symbolized by C, and thymine that is symbolized by T. Nucleotides are linked together by covalent bond to form the nucleic acid, or called polynucleotide, and this process is called polymerization. 2 nucleotides combine together or more than 2 to form the nucleic acid, or called polynucleotide. Here represents the combination between nucleotides to form a nucleic acid. This is one nucleotide bind to another nucleotide. The phosphate group is bind with the carbon atom of pentose sugar number 3, and this is the nitrogenous bases, and this is called nucleic acid, or called phosphate backbone of nucleic acid. We have 2 types of nucleic acid, do you remember? DNA, that is called deoxyribonucleic acid, and this is the shape of DNA, the structure of DNA. As you see, it consists of 2 strands of nucleic acid. RNA ribonucleic acid, and as you see, the ribonucleic acid consists of 1 strand of nucleic acid. DNA and RNA, very important substances that inside our body. They have some similarities and differences between them. First, we are going to explain the similarities between DNA and RNA. Number 1, both are formed of macromolecules that made up of nucleotide subunit. Number 2, they both have sugar pentose that has 5 carbon atoms, nitrogenous base, and phosphate group. Number 3, they both in which the phosphate group bind to the 3rd carbon atom of the previous nucleotide and form sugar phosphate backbone. These are the similarities between DNA and RNA. The following table shows the difference between DNA and RNA. Point of comparison number 1, types of pentose sugar in nucleotide. In DNA, the type is called deoxyribose sugar. It means which lack an oxygen atom than ribose sugar. But in DNA, it's ribose sugar, so it has the oxygen atom. Number 2, the nitrogenous bases. The nitrogenous bases in DNA are adenine A, guanine G, thymine T, and cytosine. But in RNA, they are adenine A, guanine G, uracil U, and cytosine C. The difference here, in DNA, the nitrogenous base is the thymine, but in RNA is the uracil. Number 3, number of strands in each molecule. DNA consists of 2 strands of nucleotides, and RNA consists of single strand of nucleotides. The location or the place that the DNA exists. DNA exists inside the nucleus of the cell, where it is involved in the structure of chromosomes. The chromosomes that inside the nucleus and carry the genes. RNA, the location of RNA, it transcribed or formed from the nucleic acid DNA inside the cell nucleus, then transfer to the cytoplasm to help in protein synthesis. The importance: DNA carries the genetic information, but RNA sensing protein which the cell needs. This figure represents the difference between pentose sugar in DNA and the sugar pentose in RNA. Here in RNA there is an oxygen atom, but here in DNA no oxygen atom. DNA consists of double strands of nucleotides, but RNA consists of single strand of sugar phosphate backbone. The 3 main types of RNA that participate in protein synthesis, and these are: number 1, messenger RNA, that is symbolized by mRNA. Number 2, ribosomal RNA, that is symbolized by rRNA. Number 3, transfer RNA, that is symbolized by tRNA. And we are going to know each one alone. Number 1, messenger RNA. First, you should know the DNA transcription into RNA. The transcription of DNA into RNA begins when the enzyme RNA polymerase binds to a sequence of nucleotides on the DNA that called promoter. So, promoters are a sequence on DNA direct the RNA polymerase to the sequence of nucleotides on DNA to form mRNA or messenger RNA. Next, the 2 strands of DNA are separated, and 1 strand only serves as a template for the formation of a complementary strand of RNA. After this, RNA polymerase moves along the DNA and joins the complementary nucleotides to the growing RNA strand one by one. The enzyme works only in the 3 to 5 direction on its DNA template, assembling RNA in the 5 to 3 direction. This figure represents the transcription of mRNA from the only strand that acts as a template of DNA. There is difference between the prokaryotes and eukaryotes in RNA transcription. There is a difference between them. In prokaryotes, a single RNA polymerase transcribes RNA, mRNA, and tRNA. But eukaryotes, there are 3 different kinds of polymerase. The messenger RNA of prokaryotes is ready for translation as soon as it's transcribed. But in eukaryotes, on the other hand, the original mRNA transcribed must be processed further in the nucleus before the mature mRNA is ready to enter the cytoplasm and participate in protein synthesis. At the beginning of each mRNA is a ribosome binding site. The ribosome binding site is a sequence of nucleotides that bind to a ribosome in such a way that the first codon AUG is positioned correctly for translation, and the last codon is called stop codon, which may be one of the three codons UAA, UAG, and UGA. At the other end of mRNA molecule, there is a polyadenine tail that composed of 200 adenine residues or nucleotides. It appears that this tail protects messenger RNA from breakdown by enzymes in the cytoplasm. This diagram illustrates the structure of messenger RNA. This is the ribosome site position, and AUG, this is the start codon, and there are three codons, they are called stop codon, and this is the polyadenine tail that prevent the breaking down of messenger RNA from any enzyme in the cytoplasm of the cell. Example number 2, or type number 2 of the RNA is called ribosomal RNA, that is symbolized by rRNA. Ribosomes are the organelles of protein synthesis. Do you remember what are ribosomes? From the inner structure of the cell, it's organelle inside the cell and help in the protein synthesis. That is consists of several types of RNA and about 70 kinds of polypeptides. In eukaryotes, the production of ribosomes takes place in an area of the nucleus called the nucleus, where several hundred thousand of ribosomes are produced per hour. Such rapid production is possible only because an eukaryotic cell's DNA contain up to 600 copies of the ribosomal. In eukaryotes, the polypeptides of ribosomes are made in the cytoplasm and then pass through the nuclear envelope. After this, enter the nucleus where the rRNA and polypeptides are assembled into ribosomal subunits. During protein synthesis, interaction occurs between messenger RNA and ribosomal RNA. The functional ribosome consists of two subunits, large and one small, one large and one small. I hope you understand the lesson today and study well, then answer some exercises to can apply what you understood by exercises. Thank you for listening and goodbye.